specifically with this question. What happens when a dosage-constrained gene is duplicated? And that will be the focus of the talk. I'll talk a bit about what I mean by dosage-constrained and what kind of duplications I'm referring to. So what's dosage-constrained gene? It's a single or low copy number gene. It's often well understood or in a fundamental pathway and well conserved. And until not long ago, they still are, I guess, the workhorses of molecular evolution. They're the infamous single copy gene that people use for very distant phylogenies. So what happens when you duplicate a dosage-constrained gene? Well, since there is strong selection against changes in dosage, you either lose it and then nothing changes, you stabilize the dosage, or you get subfunctionalization, you get novel function, localization, things that stabilize dosage per functional unit. So things go back to normal very shortly after duplication. So an example of a pathway of this type is the fatty acid synthesis pathway in plants. And very briefly, there are two copies of the pathway, one in chloroplast, shown here in orange, and one in the endoplasmic reticulum, shown in blue. And the genes I'm going to focus on are the very last ones in the pathway. And they are involved in producing fatty acids that are known to be involved in cold resistance, tolerance, and all things like that. So this pathway has been characterized in Arabidopsis originally, like almost everything in plants. And it's been found that there is one copy of the gene encoding this enzyme for ER and two copies of the gene encoding the enzyme in the chloroplast. A recent study characterized this pathway in cotton and found two copies of the ER targeted one and three copies of the chloroplast. And functional analysis shows that one of these two copies in Arabidopsis is cold-induced. There is a similar cold-induced gene in cotton, tissue-specific of this one duplicate, and low or no expression. So you can see that even though we have multiple copies, they're all specialized post-duplication. They have specific roles they fill in. These two, in both cases, are basically the steady state, the ones that are involved in the basic pathway. So we have multiple genes, but where do they come from? Since the most common ancestor of Arabidopsis and cotton, we've had triplication followed by duplication in cotton of the whole genome, and 2 rounds of duplication in Arabidopsis. So we could have one gene that end up being duplicated in both of these pathways, down both of these branches, or we could have all 3 genes here and then cotton gained a few other ones, or anything in between. So to answer this question, we can't just look at Arabidopsis and cotton. We need to fill out this space with taxa that don't share duplications. So we added data from papaya, cacao, and grape, all of which have no whole genome duplication since their most common ancestor. And they cover a pretty broad range. This node is about 130 million years old, and we've got about 60 million here and 80 million there. So we identified the corresponding genes in this taxa, and we find 3 of them in all the genomes without duplication, in addition to the ones already identified. So the most likely ancestral history is that there were 3 genes, 3 copies of this gene way back at the beginning, 130 million years ago. However, given the duplication events we know happen here, this is what we expect in terms of number of copies generated. So what happened to all of these copies? We expect 6 for each one of them in cotton and 4 for each one of them in Arabidopsis. And what we find is just a handful of them. All of this disappeared. And to address this question, we want to look also, we have the shared cold tolerance phenotype. So we want to ask, is this inherited through the same gene? Are these genes identical by descent, essentially, over here, or were they independently derived? Do we have parallel evolution here? Because we generated so many copies through the duplication that it's feasible to think that you had a novel generation of the same phenotype. So to address both of these questions, we turn to Syntenic analysis. And briefly what we did is select these three genes within their genomic background, and we picked grape, which is the outgroup, and it's been shown to have very little genome rearrangement compared to the other taxa. We extended out for about a 750 kilobase region in grape, and we looked at the presence of these regions, marked with the colored rectangles, in the other taxa. And we know we have three ancestral genes, and we indeed identify three very different genomic regions corresponding to all of them. So the expectation, having those ancestral regions, is to find, again, 6 copies of each region in cotton, 4 of each region in Arabidopsis, and so forth. And indeed, once we look across genomes, we found most of them. We found pretty much conservation for most of the regions of all 750 kbs. And a synthetic region is not the whole region. It's a series of conserved blocks in the same order, basically. So you can have growth or shrinking of the region in the different taxa. And Arabidopsis in particular has reduced its genome size significantly. But generally you find the same region in most places. There has been a large inversion, the common ancestor of cotton and cacao, that rearranged this region a bit, and cotton lost a bit of it. So now that we know where these genes would have been in most cases, we look to see where we find the existing genes. And we can place them into their appropriate backgrounds. And we can see for the diploids, they are where we expect each one goes in its appropriate background. The one thing to notice, though, immediately is that Arabidopsis has no orange background gene. Is that both of the genes in Arabidopsis are in the yellow-white background. So, looking at how the functional annotation combines with the gene presence and the genomic location, we can see that Arabidopsis and cotton have the cold-induced genes coming from different backgrounds. So it's likely an example of convergent evolution. Essentially, Arabidopsis lost the cold-induced copy if this was an ancestral state, and re-derived it. Given that we have strong selection against redundant copies, and we see 3 copies in all of the polyploids, it's likely that this feature, this cold resistance, is shared. We don't have functional assays for this taxa, so we don't know for sure. But it's likely that this feature is shared in the common ancestor and re-derived in Arabidopsis. And we looked at the possibility of Arabidopsis being just a gene conversion event, given that both of these chloroplast target regions are relatively similar. You could have a crossover event that moves a gene from the orange background into the yellow background. And we have found that these 2 are indeed sister genes, and in fact, they are likely derived after that duplication of that. So to summarize, this gene family is indeed dosage constrained, which is not a surprise given that it's a fundamental pathway. And subfunctionalization or loss follows duplication. Of all the 6 or 4 copies we expect, we'll see at most 1 or 2. Moreover, the cold-induced phenotype is likely shared by many plants, and the Arabidopsis gene, which has been the model for the cold-induced gene for this pathway, is actually a newly derived phenotype, it recruited a new gene for this job. So that's really the whirlwind tour of this project. And what we're trying to understand in terms of how does the dosage constraint work, and what happens to specific functions when you go through so many rounds of duplication plus dosage constraint. With that, I would like to thank the people that helped, or in many cases did all this work. The Gore lab, which I'm currently working with for now, the USDA and BYU folks that did most of the cotton characterization work, which is currently being prepared for submission, and the funding agencies, Cotton Inc. for the work in cotton and Cornell University. With that, I will leave up a picture of what actual Syntenic analysis looks like, just because it's colorful, but it's a nice background